Yes, now YouTube has started, and only 30 seconds. You give me 30 seconds, Prushanjali.
Yes, sir.
Wait for 30 seconds only, because I will
Hmm.
share this YouTube
Yes, sir.
first.
Okay, sir.
I have shared my YouTube with the Prushanjali, and Prushanjali,
Yes, sir.
you can start.
Okay. Thank you, sir. Good afternoon to everyone. I welcome you all on a PhD webinar series. Today's our speaker is Dr. Anupriya Chudiwal ma'am. Uh, Currently, she's working as assistant professor in Institute of Bioscience and Technology at MGM University, Aurangabad. She has 11 years of teaching experience. She had published 7 research articles in reputed journals. Also, she has participated in various seminar conferences and workshops. I welcome you, ma'am, on behalf of our Microbiology Society. Now I request you, ma'am, to start your presentation.
Yes, thank you, ma'am.
Thank you, ma'am.
Good afternoon, everyone. Is my slide is visible to you, ma'am?
Yes, ma'am.
Okay, thank you. Good afternoon, everyone. First of all, I'm very thankful to the President, Dr. A.M. Deshmukh, sir, Microbial Society of India, and organizing committee to give me a valuable opportunity to present my PhD work in front of you. Thank you. With the permission of sir, I'm going to start my topic. So,
Yes, proceed.
thank you, sir. But myself, Dr. Anupriya Chudiwal, I'm working in IBT, MGM University, Aurangabad, and my research topic is bioprocess development for production of exopolysaccharide from marine bacteria and evolution of its antimicrobial potential. And this work was done under the guidance of Dr. Smita Dharmadikari and Peshwe ma'am. So what is the need of research? At a present, the microbial resistant to the antibiotic is becoming a serious problem. Day by day, pathogens are becoming resistant to the antimicrobial agent. Multi-drug resistant bacteria have emerging trouble in the world of medical treatment. So, the large number of antimicrobial compounds again are narrow spectrum. They are sensitive to acid hydrolysis and they kills the normal flora. Hence, there is an urgent need for the novel and alternative source of antimicrobial agent. Development of new exopolysaccharide having antimicrobial potential can be used in any combinatorial therapy. The most uncommon unavoidable obstacle of wound healing is the installation of an infection. Due to extremely difficult system of wound healing, effective and targeted cures are still needed. Hence, the research is currently provoked to find a more efficient therapy. Traditionally, the wound dressing is applied to shield the wound from external contamination. But nowadays, these dressings can be functionalized with many antibiotics and some EPS that is having antibacterial properties. Again, the constitutive production of EPS from marine microbes is very low. And owing to its vivid application, there was need to explore EPS as an alternative source for antimicrobial agent and to develop economic production and ultimately their successful utilization. So, what is the significance of research? To explore this marine microflora for the production of exopolysaccharide and evolution of broad spectrum antimicrobial activity against pathogen. The synthesized exopolysaccharide used as a broad antimicrobial to overrule the problem of multi-drug resistant and control the other detectable infection using this synthesized EPS. Next, what do you mean by the exopolysaccharide? Exopolysaccharide is a metabolic product which is secreted by microorganism. These EPS contain high molecular weight polysaccharide, which are ranging from 10 to 13 kilodalton. Different types of exopolysaccharide are produced, like from plants, algae, bacteria, and marine bacteria also. Here we work on marine bacteria, as marine has remained unexplored with respect to its microbial diversity and its application. And EPS from marine bacteria are generally heteropolysaccharide in nature, which contain 3 to 4 different monosaccharide units, and components that are most commonly found in marine EPS are pentoses, hexoses, amino sugars, and the uronic acid. So microbial exopolysaccharide has got a number of applications, like in field of food industry, health and medicine, and various industries. But in our research, we are mainly focused on our aim or application in relation to the antimicrobial activity, and the whatever the EPC was synthesized, they are used as an wound dressings. So this is the main aim and objective of my research that I have done. First, it is isolation, screening, and identification of potential exopolysaccharide producing bacteria from marine samples. Then optimization of exopolysaccharide production by means of one variable at a time approach. Then some statistical optimization method are used, like blanket woman design and central composite design. Then the scallop studies and the strain improvement studies were done. After that, production, extraction, purification, and characterization were done. And lastly, the application of exopolysaccharide was done in relation to its with terms of antimicrobial activity, antifungal activity, wound dressing film, their MIC and MBC studies, and other application of EPS were also studied. So my research work was done in 3 phases: phase 1, phase 2, phase 3. The phase 1 is about isolation, screening, and identification. For isolation of marine bacteria, total 12 different coastal area of sample were collected from 15 centimeter of a depth, and it was procured in lab. This is the table which shows physicochemical properties with respect to pH, temperature, turbidity. This is the pie chart which showing the percent distribution of isolate, and we got a number of marine microbial diversity in Alibag beach, followed by the Marur Jinjira beach. So this is the isolation phase. After that, as our aim was to produce exopolysaccharide which having antimicrobial activity, for that a different screening strategies were applied. This screening strategies that is used in primary screening was the string test, congruent agar plate test, mucodal nature of the colony. Here, that is the whatever the 160 bacteria were isolated, they were further screened for these 3 tests. And this is the plate showing the mucodal nature of the colony. And as there is a production of exopolysaccharide, this congruent agar plate turns to be a black as it is synthesizing the polysaccharide. Again, this is the prominent string test was observed upon lifting the loop after touching to the colony, which indicating the positive result. So all these 39 bacteria, they are further screened for the secondary screening, and secondary screening were done in terms of their qualitative as well as quantitative characterization. So in qualitative characterization, the Molish test was performed, and this is as there is a purple wallet ring, it gets formed between these 2 junctions, indicating the presence of polysaccharide, and it was again confirmed by the Anthron test. This is the qualitative characterization which done for the 39 isolate, and after that all these isolates were studied in their quantitative way, where we EPS get extracted, and we get a dry weight, or we take a dry weight of EPS, and it was based on their dry weight of exopolysaccharide. It was further classified into the 4 categories, that is the weak EPS producer, mild EPS producer, moderate EPS producer, and the strong EPS producer. Strong EPS producer are those which produce 0.95 gram per liter EPS, which is more than the 0.95. So only the 7.69% were identified as a strong EPS producer, and this strong EPS producer are the 3 EPS producer, that is the AB4, which gives the yield of EPS that is 1.4 gram, while MZ10 it is 0.98 gram, and N8 it is 0.96 gram. So these 3 categories were considered as a strong EPS producer, and it was further analyzed. So this is the secondary screen isolate, where we found that their morphological characterization were also done, and it was found that the AB4 and NZ, these are the gram negative, while N8 it is the gram positive. So this is the biochemical characterization chart, and this is a table which showing biochemical characterization of secondary screen isolate, which is tested, which is done for AB4 bacteria, MZ10 bacteria, and N8 bacteria. Again, the enzymatic profiling of all these 3 isolates were carried out, and this is the slide which showing the which reveal the AB4 isolate, which showing all the tests positive except the starch hydrolysis test. Again, the tertiary screening was carried out on the basis of their antimicrobial potential of EPS, which is produced from these 3 selected isolates. So for the same, EPS which was produced by these 3 isolates was tested for its antimicrobial potential against 8 MTCC pathogens. So these are the 8 MTCC pathogens that I have taken from MTCC, and here a result revealed that the AB4 bacteria, this is the AB4 bacteria which is giving a more prominent zone of inhibition as compared to others. So these AB4 bacteria, therefore the AB4 bacteria were further selected for further studies. So for that these AB4 were identified, and for that the VTEC system of identification along with 60SR RNA sequencing were done, along with some supplementary result of biochemical tests that I have performed in our lab, and it was identified that it was the Pseudomonas aeruginosa, and this is the genomic DNA and the PCR reaction where I got this result. Again, the genotypic analysis was done, and the sequence was deposited to NCBI gene bank with accession number MH324730. This is the BLAST result and the phylogenetic tree that I obtained for my organism. So this is all about the phase 1, where isolation, screening, and identification was done. After that, the second phase is about the optimization and the scale-up studies. For that, this is the methodology that I have done. Some physicochemical characterization and the formulation of media were done. So optimization of process parameter by one at a time factor, it was done to enhance the EPS production by the AB4 bacteria. So to determine the base cultural conditions, stepwise modification of the production medium was done that influenced the EPS production. For that, the cultural parameter as well as nutritional parameter were considered. In cultural parameter like the culture media, inoculum size, incubation time, agitation speed, pH and temperature were considered, while in nutritional parameter, as my bacteria were from the marine, so I have to check the salt concentration, carbon source, nitrogen source, and the metal source. So this is the result of one variable time approach, where the growth curve studies of AB4 revealed that the maximum EPS production was achieved during the 72 hours at a stationary phase with a yield 1.25 gram per liter. Again, the ability of AB4 to produce exopolysaccharide in different culture media was assessed, and for that six different culture media were selected according to the literature survey, and it was found that the maximum EPS production was found in YMG broth, that is the yeast extract malglucose broth, followed by the marine broth, which is supplemented with 1% glucose. Again, here the 1% inoculum volume with a 72 hours incubation gives the maximum EPS production. Again, pH is one of the important factors for the growth of microorganism, and maximum EPS production or maximum EPS production was observed at pH 10, which itself defined that the microorganism can be grown at a stress condition, that is the pH about a 10. Again, 30 degrees Celsius is optima. It is found very suitable for the EPS production. Next, again, our results show that EPS production was increased as the salinity was increased, and maximum EPS production is found at a 3.5% NaCl concentration with a yield 4.7% or 4.7 gram per liter. Again, agitation is extremely important for proper mixing and the aeration of the media. So the increase in the yield along with the agitation emphasizes that the obligate aerobic nature of bacteria. But as we didn't get so much satisfactory result when we compared with the static one. So agitation speed at a 100 RPM and the static one, it is similarly somewhat same. So we didn't get any very much satisfactory result with agitation. Next one, the nutritional parameter were checked by one variable at a time factor. An EPS synthesis is mostly depend on the carbon availability in the culture media. So to find out them, the glucose, we have tested 11 different carbon sources, and among them the glucose gives a maximum EPS production. So here, that is the glucose gives a maximum EPS production with a yield that is the 3.3 gram per liter. And here a simple nature, easily available, anechoic nature of glucose overruled the other sugar, and again it is very suitable for the industrial applicability. Again, the maximum EPS production, that is 3.6 gram per liter, it was obtained when the media was supplemented with the ammonium sulfate. Among the tested metal ion, the maximum EPS production was observed in a magnesium sulfate followed by the sodium hydrogen phosphate. So this is a summary of one variable at a time approach. The one variable at a time experiment show the following variable at optimum variable for EPS production. From the one variable time approach, again, there is no interpretation of intra-variable type. So here, to reduce the complexity of a process variability or to reduce the production time, a statistical design were used by evaluating the main large effect to improve the production yield and to get a closer confirmation of output response. So for media optimization, a 3 statistical design like Plankett-Bowman design followed by strip and test and experiment and the CCD were performed. Here PBD, it reflects the relative importance of various factors that are used, and it gives significant factors which are affecting the response. Again, the strip and test and experiment, whatever the result that are obtained in PBD, it was followed to the strip and test and experiment, and this strip and test and experiment, it tell us or it move rapidly towards the neighborhood of optimum response. CCD provide an optimum value. This CCD provide optimum value of individual factor to get exact composition of a media. So the nutrient that are selected or nutrient that are dominant in one variable at a time were selected for further optimization by the PBD, and total 12 variable like these include the 4 carbon sources, 4 nitrogen sources, and 4 mineral sources with their high value and the low value. And this is the Plankett-Bowman design. For that, the Minitab software were used, and it gives a 60-run experiment, and this is a design which is given by the Minitab software. So among here, this is the Minitab software design where we got the maximum EPS production in run number 6, that is the 3.4 gram per liter, and it was nearer to our predicted value. So this is the effect and the coefficient of EPS production with PBD, where we know that glucose, the C, it is glucose, then the ammonium sulfate, then magnesium sulfate, and the sodium hydrogen phosphate have a confidence level more than 95% with a p-value that is the less than 0.05%, 0.05%, which prove our model is significant. This is the graphical representation of ANOVA, which is known as an Pareto chart, and this is the reference line that show that this glucose, ammonium sulfate, sodium hydrogen phosphate, and magnesium sulfate, it has a significant effect. So these four nutritional parameter, it was whatever the significant variable that obtained in PBD. From that we designed the stipend-ascent experiment, and from the result of stipend-ascent experiment, we found that the run number 5 gives yield that is 4.1 gram per liter EPS. So here that is the fifth run design. So for that, the fifth run design were utilized as a center point for designing the next statistical design, that is the CCD. Which gives a exact value of variables. So here the next design was done with a CCD, that is central composite design. For that, for CCD designing, again we consider here the low value and the high value based on the stipend-ascent experiment data and the design expert 18. For that, for PPD, the Minitab software were used, and for this central composite design, a design expert 18 were used, and which provide a 30-run experiment. So this is the 30-run experiment which is given by the software 18. And according to that design, we carry out the experimentation. We extract the exopolysaccharide by the chilled ethanol precipitation method, and we come to know that the run number 15, it gives the maximum yield of EPS, that is 4.53 gram per liter. This is the ANOVA result, which said that the model is significant, and lack of fit, it is a non-significant. So this is ANOVA result. Here the design expert itself provide a validation experiment model, and according to the confirmation report which is given by the software, we got that this level for the glucose, ammonium sulfate, sodium hydrogen phosphate, and the magnesium sulfate. And after experimentation using this level, we got the yield that is 4.53 gram per liter, which is nearer to predicted when which is predicted by the software, that is the 4.52 gram per liter EPS, and it was shown that the model is get validated. Again, this is the graphical representation of the 3D response surface plot and the 2D counter plot, which is given by software, and where which indicate that there is a proper interaction which is occur between that variable that are chosen, like the glucose, ammonium sulfate, sodium hydrogen phosphate, and the magnesium sulfate. This is the normal residual plot. In this, all points were come to on a straight line. This is which confirm that there is a proper utilization of nutrient at optimum level, and it proves design was more accurate. So this is a comparative account for the basal media and the modified media. So YMG broth, it is an basal media. After that, we with the optimization method with the CCD, we got this modified media. And in modified media, we got that there is a 1.5 fold increase. It was obtained after optimization process. Again, the antibacterial activity against S. aureus was checked with the basal media, with the EPS which is extracted from the basal media, and the EPS which was extracted from the from this YMG medium, and it was found that the zone of inhibition are also increased. So the yield was increased about the 1.5 fold. So again, after that, to see the effect of mutagen on the strain AB4, in next phase, the strain improvement studies and scale-up studies were carried out. And to see the effect of mutagen on strain improvement studies were carried out by considering this physical, like UV, and the chemical mutagen. and some scale-up studies with relation to their inoculum size, vessel volume, effect of various solvent. Again, optimization studies, it is carried out. First, the optimization studies was carried out for the media optimization, and in second thing, this optimization was done to extract the EPS. So some extraction parameter were get optimized and fermentation were checked. So here in physical mutagen, the UV mutagen, UV radiation from 2 to 20 seconds were used, and their LD50 was calculated, and it was found up to that 12 seconds is an LD50, and those colonies were considered as a mutant which survive above 50% survival, and their EPS production was checked and randomly selected UV mutant 11 used the yield that is 4.1 gram per liter EPS. A second mutagen were used, that is the sodium azide, and the concentration from 10 ppm to 100 ppm were used along with the control, and their LD50 was found to be a 15 ppm, and the colonies with change in their morphology and above LD50 were selected, and their EPS production was checked, and the colony number 7, that is the SAM7, show the improved EPS production than wild type, and other, that is the 5.5 gram per liter EPS was obtained. And we are not getting any encouraging result after trying several combination of this ETBR. So the SAM7 mutant of AB4, it was further used for further study. Again, the inoculum volume was found to be a 1%. Our results show that the 1% inoculum volume gives maximum yield of EPS. Scale-up studies were done with optimized media, and it was found that the EPS production was consistent when there is a increased volume of media. So here I have increased the volume from 50 ml to 20 up to the 1000 ml, and it was found that there is a consistent yield. Again, extraction during downstream processing, a different solvent were used, and these different solvent were used to extract the EPS, and it was found that ethanol gave a more yield of EPS. So here ethanol is used as for the ethanol precipitation method. So next, that is the optimization study with the extraction process. So here a 6 different variables. So further optimization of EPS extraction process were done by statistical method where PBD and CCD were used. Here is 6 different variable like ethanol percentage. So for to extract the EPS, the ethanol percentage, ethanol multiple, how much amount of ethanol we are requiring, then precipitation temperature, precipitation time, drying temperature, and the pH of your supernatant were considered. So these 6 different variables were considered, and ANOVA result shows that ethanol multiple, see here BCD, that is the ethanol multiple, ethanol precipitation time, and precipitation temperature. These 3 variables are more significant. So these 3 parameters were used, and which had a significant influence on EPS extraction. Here in Pareto chart, they show that their p-value is the less than 0.05 p-value was obtained. Therefore, these 3 parameters were significantly taken into the consideration. Then these 3 parameters were considered for next statistical design, that is the central composite design, where it gives a 20 run experiment, and in that the 60 run experiment gives yield that is 6.49 gram per liter EPS, and this is the ANOVA result where it shows that the model is significant and the lack of fit, it is a non-significant. Here again very high value, and the correlation coefficient, that is 99.85% indicate that there is a good agreement between experimental and the predicted values. This is the graphical representation which is given by the design expert 18, and the this is a 2D counter plot, response surface plot, and the 2D counter plot. Again, the optimal extraction parameter for ethanol, it is a 5.33 ml. Precipitation temperature is it is a 4.2 degree, and the precipitation time, it was required about the 12 hours 32 minutes. So this is the three extraction parameter were get optimized by this method. This is the normal probability plot was satisfied as the residual plot is approximately on the straight line. This is the comparative account where in basal media we get a 3.2 gram per liter EPS. After that, after optimization of media in modified YNG broth, we get a yield of 4.5 gram per liter EPS, and after extraction process, we got a yield that is 6.49 gram per liter, which is nearer to your predicted one. So again, the batch fermentation studies and feed batch fermentation studies were done to check which fermentation was suitable for EPS production, and it was found that the feed batch fermentation was in feed batch fermentation a 30% rise is obtained. So this is about the second phase, that is the optimization. After that, the production. After that, the third phase was done with production purification application studies. This is the methodology that I have done. So here the production was terminated after the 72 hours at 30 degrees Celsius, and it was purified by the ethanol precipitation method, and it was again purified by the deproteinization with trichloroacetic acid, and it was dialysis, and finally the DAE cellulose chromatography were done. And while doing the DAE cellulose chromatography, the elution profile of EPS was checked, and it was found that the EPS was started eluting at fraction number 2, and it was maximum at a fraction number 5, and its concentration was determined. All these concentration were determined by the Dubos method, that is the phenol sulfuric acid method, and all this phenol sulfuric acid method were checked, and here the purification profile of EPS was calculated, and it was found that after ion exchange chromatography with the EPS, the 21.51 purification fold was increased, and with a yield of 27.81%. After that, the characterization of EPS was done by the conventional method, where the conventional method like heat shock studies, cold shock studies, pH, and the stability of EPS was checked. So here the activity of exopolysaccharide AB4 was analyzed at varying temperature ranging from 40 to 110 degrees Celsius, and our results show that the maximum activity is up to the 70 with a marginal with a 60 degree with a marginal with a very low marginal decrease activity, which is very beneficial in any food industry. Again, the cold shock studies were revealed that the 4 degrees Celsius was suitable for the storage condition. And again, the pH stability was also checked, and from the study of pH stability, it was found that our EPS worked at both condition, that is the at a neutral as well as at an alkaline condition. So it was stable, and it was stable at both the condition. Again, the stability here, AB4 exopolysaccharide was found to be quite stable with a marginal decrease at a 6% up to the 6% and 8% after 15 to 30 days up to. Then some characterization study or some characterization of biomolecules were done, and it was done here the monosaccharide, whatever the here monosaccharide which present monosaccharide which you were present in that produce EPS was checked by acid hydrolysis followed by their thin layer chromatography, and it was found that the glucose and the glucose and mannose is the main component. Again, some analytical methods to study carry out the chemical characterization, like some carbohydrate and small concentration of protein as well as the uronic acid were analyzed, And for the total carbohydrate concentration, phenol sulfuric acid method were used. For protein, Pauling-Lowry method it is used, and for uranic acid, the carbazole method it is used. And this is our result, that is the Pseudomonas aeruginosa IB4, the present study, and it was compared with the others. Again, the UV visible spectral studies were determined, and UV visible absorption spectrum of EPS had shown maximum absorption at a 210 nanometer with a very low, very less shoulder around 262 to 80. Again, FTIR studies were done. This is a graphical representation of FTIR, and these FTIR were used to examine the vibration and the functional nature of the molecule. And this FTIR spectrum of EPS display a strong broad band at a 3624.15 centimeter wave number and 307309, revealing the presence of OH stretching. Again, the signature peak was obtained at a 900 signature peak of glucuronic acid residue were detected at a number 94846, which is very close to the alginate spectrum. Again, this OH stretching, CS stretching, indicating the existence polysaccharide nature of the molecule. Again, next, that is the after FTR, the stability and the longevity of a polymer are usually determined by the thermogravitometric analysis. And to see the thermal transitions which is occurring in EPS, for that the differential scanning calorimetric DSC were performed, and from the DSC curve the exact degradation of temperature was found to be 247.42. So it's good thermal stability, may be ideal candidate for both pharmaceutical as well as food applications. After that, the HPLC analysis of active fraction of AB4 showed that there is a single peak. It is obtained at a retention time 4.87, indicating the homogeneity of polysaccharide. This is the GCMS spectrum, which is given by the exopolysaccharide AB4 EPS. Again, besides chemical property, some application of exopolysaccharide is mainly depend upon their rheological behavior. So their rheological characterization was done, and rheological property was determined in lab by the Ostwald viscometer. The EPS show a good rheological property at alkaline condition, that is the with a good MPS at a 9. And again, at alkaline condition up to the 90 degrees Celsius with 125 MPS. Again, there is a presence of monovalent, and the divalent cation also increase the viscosity. Here some monovalent and the divalent cation were used, and they show that they increase the viscosity of PPS solution as compared to the control. And which indicate its suitability in food and the cosmetic industry as a thickener or the viscosifier. Again, some quality control studies were done, and these quality control studies were done by means of membrane filtration sterility test, and it was observed that there is a no growth was observed, no antimicrobial growth was observed on selective nutrient, selective culture media, which is used after the membrane filter sterilization of EPS. Again, some cytotoxicity tests were done, and this cytotoxicity test was determined against viable lymphocyte by the trypan blue assay, and there is a no significant cytotoxicity was observed against the lymphocyte. Again, this third, it is regarding to the application. So as my main application is in relation to the antimicrobial activity. So for that, some clinical pathogens were collected, and their registotyping analysis were done in a lab. Along that, some fungal plant pathogen were also isolated. So here some clinical pathogens, like 3 from the UTI, that is the urinary tract infection, and 7 from the burn patients were procured from the pathology lab, Jekta pathology lab, Barshi, and successfully characterized in a lab, and it was further identified in our lab, and it was identified that These are the some clinical pathogens, and these clinical pathogens were tested to check the antimicrobial activity of my EPS. Again, some and their register typing were done, and their MAR index was calculated, and MAR index was found to be MAR index of antibiotic for secondary wound infection causing pathogen, that is the burn pathogen, were ranged from 0.04 to 0.085, while the uropathogen was 0.23 to 0.25. And if MAR index it is greater than 0.2, which indicate that this organism it originated from the environment where the several antibiotics were used. So their antimicrobial activity was checked, and this is the slide. Here, this is a slide which showing the isolation and identification of fungal isolate, like the Aspergillus niger were obtained, then the Fusarium, Aspergillus flavus, Fusarium, and one pathogen it was taken from the NCM, that is the Candida albicans. Again, this exopolysaccharide was checked. Their antimicrobial activity was checked against some NTCC pathogen, some fish pathogen, and some reference pathogen that are procured from the pathology lab, and some clinical pathogen. So these are the pathogens where I have checked the antimicrobial activity of my EPS, and here the exopolysaccharide show notable antimicrobial activity against all selected MTCC pathogen except Pseudomonas aeruginosa, which is giving no zone of inhibition. Again, the maximum antimicrobial activity was observed with a 24 zone against the Azorius. Some fish pathogen are also tested, which also gives the and show the zone of inhibition when it was tested with the exopolysaccharide AB4. Again, some this is the slide where sorry, this is a slide where some these are the slide where the MTCC or known pathogen were tested. This is the some clinical pathogen that are isolated from the UTI and the some burn patients, and it was checked, and against same result was obtained here, that there is a no zone of inhibition was obtained against Pseudomonas aeruginosa, and while some bacteria were get these are some bacteria where the zone of inhibition, more prominent zone of inhibition were obtained. Again, the exopolysaccharide film was prepared according to the Anna et al. And their porosity, their swelling ratio, and their homeostatic and in vitro biodegradation evolution were done. So this is the EPS film. This is the EPS film composite bandage were prepared in a lab, and it was further characterized. This EPS composite bandage, it was like it showed the porosity, that is the EPS composite bandage. It is a giving a porosity that is 73.89% while compared with the control. Again, the hemostatic potential are also good, and swelling ratio, EPS composite bandage, it gives 20% swelling ratio as compared to the control, that is 24%. Again, these film were used for the this film were used in vitro for their antimicrobial activity. So here the EPS film composite EPS film has drawn attention in application of wound dressing due to their hydrophilicity, porosity, biocompatibility. So we have observed in vitro antimicrobial activity of EPS against both gram-positive as well as the gram-negative organism that are commonly cause secondary infection in burn patients. So some clinical pathogens were checked, some burn isolate that are procured from the pathology lab were checked, and it was found that the Azorius here, that is the Enterococcal cloacae, then Enterococcus faecalis, they are showing the zone of inhibition against that EPS film, and it was controlled with a sodium alginate film over there. Again, along that, the other applications were also studied, antifungal activity. It was checked against the plant pathogen, and here notable inhibitory effect was found against the Aspergillus niger, Fusarium species, Penicillium species, while hexapolysaccharide was not able to show an inhibitory effect against Candida albicans. Along with some antimicrobial activity, other applications were also studied, like uh, uh, this this exopolysaccharide, it used as a probiotic for the aquaculture. So as it was inhibiting the fish pathogen, that is Aeromonas veronoli. So this Pseudomonas supernatant, it can be used as a probiotic for the aquaculture. Again, the antioxidant studies were done with a DPPH method, and it was also found to be suitable. After that, some other applications were also studied, and other application like the EPS based curd has been made, and here the attempt has been made to prepare the EPS based curd. The curd was prepared using the cow milk and fermenting with a native inoculum along with 1% EPS. So inside B, inside B, the whatever the EPS it is present in that native culture plus purified 1% EPS were added, and it was found that its physical chemical properties of a curd was checked in relation to their texture, water holding capacity, and the pH and their viscosity. And the water holding capacity of a set B, where the EPS was added, it was good with a 4.16 centipose, having a smooth with a thick consistency, and having the viscosity of 4.16 centipose as compared to control. And here the low pH responsible for contributing to the texture of curd, and it also ionizes some calcium that facilitates the absorption in the intestine. And some emulsification studies were also done, or emulsification index activity was also calculated against various oil, and the maximum emulsification index was observed in sunflower, groundnut, and the mustard oil respectively. So lastly, the conclusion of my research topic is that I got a novel Pseudomonas aeruginosa IB4, which produced high exopolysaccharide with antimicrobial potential, and it was from marine source. The optimized economic production through one variable at a time, and the multivariant mathematical approaches with 5.07 fold increase. Again, the strain improvement through sodium azide treatment found better than the wild type. It was validated the efficiency up to a scale up and the purification. Then their structural studies were elucidated with the FTIR, HPLC, thermogravimetric analysis, and GCMS. Their antibacterial activity of EPS and EPS film against number of tested microbes, including MDR. And this research work pursued the economic production and the earnest application of marine bacteria AB4 EPS as an alternative, efficient antibacterial EPS producer. So this AB4 EPS was applied for antimicrobial agent against MDR pathogen. EPS based film to prevent wound infection. It is used as an antioxidant agent, probiotic for aquaculture, and EPS based curd and the emulsifying agent. So to the best of our knowledge, this is the first report to demonstrate in vitro antimicrobial dressing of bacterial EPS produced from marine source. The current study supported the potential candidate of EPS with additional pharmacological research in medicinal and as well as in cosmetic formulation. So thank you all.
Excellent, Anupriya.
Thank you, sir.
Yes, you have done a means actually I'm happy that you have participated on our platform.
Thank you, sir. This is thank you for giving me an opportunity. only the one call is came from Nitin sir. I said that yes, sir hai to main kar lungi.
Correct.
And
That's
sir,
very good.
now I'm working in IBT.
Yeah, I came
Institute
to
of
know. Yeah.
Bioscience Technology.
Yes, very good, very good.
Yes,
So I
sir.
will visit sometime to your institute.
Yes, sir, definitely. Thank you.
Yes. Now, Krishanjali, proceed.
Yes, sir. Thank you, ma'am. Thank you very much for your presentation. Now we'll move ahead with the question answers.
Yes, definitely.
Participants, please, if you have any questions or doubt, please text in chat box.
They can ask question in chat box or they can
Directly, yeah.
also directly by unmuting themselves. I think you did your partial work under Dr. Dharmadhikari.
Yes, sir.
Major work.
All my thesis was submitted in her presence. After that, due to the COVID situation, and then this happened, and after that, only the Viva part was done with Dr. Peshpe ma'am. And whatever the PPT presentation which were held during Viva, so she helped me a lot of along. You also helped me a lot of because my some report was not came and all that thing. So thank you, sir, for that. At that time, your support is very beneficial to me, and I moved from that thing.
Yes. No, everything is for it was our fate, and we have to do many things in the future in the subject. Now, Dr. Prakash Halami want to ask something, please.
Yes, sir.
Dr. Halami, sir, please, you can ask. Dr. Halami.
Good afternoon.
Good afternoon, sir.
Very good work. Actually, network issue is there with my.
Okay.
1 minute.
Just wanted to, because we are also working on EPS as
Halami,
a emulsifier.
Okay.
Can you hear me, sir?
Yeah, 1 minute. Let me tell Anupriya.
Yes, sir.
Halami sir is a very eminent scientist from CFTRI Mysore.
Okay.
Thank you, sir. Thank you.
You are fortunate that he listened your lecture completely.
Thank you. Yes, sir.
So he is having one difficulty. Please, Halami sir.
Yeah. So as far as the food application of EPS, very wonderful work what she has demonstrated, and we are also using the EPS for thickening agent in several of these juices. So from that angle, it was quite useful. Only I wanted to know whether this marine bacterium, what you have characterized, is it safe?
Has? What, sir?
How safe is that? The marine bacteria which you have characterized, is it a safe?
Is it a safe?
I have checked their cytotoxicity test after extracting the EPS from my organism. I have checked against the lymphocyte, and there is no any significant activity was observed against that lymphocyte. So
if you are going to use the EPS, then it has to be purified to homogeneity, because if organism is not safe, just like from E. coli, whatever protein we use for a so for food application, when we use, we actually ensure that the organism which is producing it also should be the safe or gross status.
Yes.
Another one point in your conclusion, actually, it should be the prebiotic activity of EPS.
Probiotic activity for the aquaculture, sir. It is not
probiotic activity of what?
Probiotic activity for aquaculture, like as my EPS was inhibiting the fish pathogen. So in aquaculture, if I'm giving this EPS, so whatever the fish pathogen, due to that the fish were killing and all that thing, so it is supporting to that aquaculture in that regard, sir. And there is one literature where the this EPS of Pseudomonas aeruginosa were used as an probiotic for that aquaculture. It is not for the humans.
No, as a prebiotic, it should be a prebiotic activity.
Okay. Yeah, sir.
Very good.
Thank you, sir.
Okay, if there is no any question, Prashanjali,
Yes,
you
sir.
can conclude.
Now we have came to the end of our webinar. It's time to thank you everyone. First of all, I thank you, Dr. Anupriya Chudiwal ma'am, for her wonderful presentation. Also, I thank you, Dr. M. Deshmukh sir, President of Microbiology Society, and all other participants who made this meeting possible. And thanks again for joining us today, and hope to see you again next time. Thank you, everyone.
Thank you, ma'am. Thank you, sir.
Sir, I will end the meeting.
Yeah, thank you. Thank you, everyone.
Okay.